So welcome back from the break. Let's take a look at these concept check questions and then we'll go on and talk about gastrulation and organogenesis. So briefly describe how the fertilization envelope develops in sea urchins. What function does it perform? Well, remember the fertilization envelope develops from the modified vitelline layer and the jelly coat. So those two layers are initially anchored to the surface of the egg cell by the sperm compatibility protein receptors that are in the jelly coat, or the receptor portion is in the jelly coat, but it then penetrates through the vitelline layer and anchors into the cell membrane to hold everything in place. When the cortical reaction begins, the digestive enzymes, or some of the digestive enzymes from the cortical, split the receptor proteins, releasing the vitelline layer and the jelly coat from the anchored position adjacent to the egg cell membrane. As they float away, as a result of water being drawn in by the osmoticum into the perivitelline space, other hydrolytic enzymes then modify both the vitelline layer and the jelly coat to harden them against the acrosomal enzymes so that they are no longer digestible by the acrosomal enzymes. So the combined effect of floating the vitelline layer and the jelly coat layer away from the cell membrane, creating space between the cell membrane and this combined structure, and the chemical modification of that combined structure to harden it against digestion by acrosomal enzymes, results in the formation of the fertilization membrane. And remember, that is triggered at the point of sperm penetration into the egg cell and moves in all directions away from that point around the outside of the egg cell, releasing the proto-fertilization envelope from the cell membrane and ultimately merging at the other side with a complete release of the proto-fertilization envelope. What function does the fertilization envelope form? Initially, to block access to any more sperm, but ultimately also to act as a protective barrier around the early embryo as it begins cleavage and on into gastrulation via the blastula. So let's look at the second one. What would happen to an unfertilized sea urchin egg if you injected it with calcium ions? It would undergo the cortical reaction. We would see triggering of the activation of the egg. We would see the formation of fertilization envelope, but we would probably not see cell division because we only have a haploid egg, and we would probably not get development of the mitotic structure. So we wouldn't expect it to begin mitosis, but we would certainly expect to see it activated to undergo the early steps of fertilization and egg activation. So let's move on and take a look at gastrulation and then organogenesis. So gastrulation, as I've suggested several times now, is probably the single most important of the 4 steps, because here you're going to completely rearrange the blastula into the free germ layered embryo, which will ultimately develop the organ systems in organogenesis that are found within the body of the developing organism. At some point too, particularly in organisms where there is very little or no yolk, a primitive gut referred to as the archenteron will develop. It may or may not be functional. So if you've got an organism like a chicken, where you've got plenty of yolk to support the development of the embryonic chick, and it's doing so inside a hard-shelled egg, then obviously the gut can't function as a proper feeding organ, but it can still break down material released from the yolk and provide nutrition to the developing embryo. And we do get the development of this primitive gut, this archenteron, as a part of the gastrulation event. So as we look at gastrulation, we should be on the lookout for these events occurring, and we'll see how in different types of animals these events play out. So the first thing to recognize in all of this is that, again, we need to take into account that there are two different types of symmetry. There is the radial symmetry of the cnidarians, the jellyfish, and the sea anemones and the like, coral polyps, and of the ctenophorans, which we don't talk about very much, but I'll introduce them to you when we do chapter 33. And then there is the bilateral symmetry that we recognize in most other animals. But when we go to the bilaterals, we also recognize that there are two different groups of bilaterals, one of which is a valid clade, the so-called deuterostomes, and one which is no longer considered to be a valid clade, the protostomes. And we'll talk more about that when we get to chapter 32, which is our next chapter, but which we still recognize as a descriptive term because it relates to what happens to a structural feature that occurs during gastrulation called the blastopore. This is the first opening into the primitive gut, the archenteron, that develops. And what that first opening develops into determines whether you're looking at a protostome or a deuterostome. So first of all, let's just take a look at a simple diagram that illustrates what's going on during a simple gastrulation step, gastrulation in something like a sea urchin. Now, what's interesting here is we need to remember that sea urchins are echinoderms, and echinoderms, as you'll discover next week in lab, are members of a clade that is considered to be a sister clade of our own, the chordates, very, very closely related. So even though the external appearance of something like a sea urchin or a sea star would belie the fact that they're closely related to chordates and vertebrates, they do after all show a radial symmetry, we show a bilateral symmetry. We do recognize echinoderms as being bilaterals because they show all the early developmental stages of bilaterals. They also show the early developmental stages of deuterostomes, which is the same early developmental stages that we show. However, they undergo a major metamorphic event that changes their body plan from bilateral in the larval stage to radial in the adult stage. But because they show the bilateral / deuterostome larval features, they are considered to be bilaterals. So they show some of these early stages. So this shows the external features, this shows the internal features. And of course, you've seen this in the embryology lab that you've already completed. So how does this symmetry play out in terms of gastrulation? Well, it turns out that the number of germ layers is correlated to symmetry. So the cnidarians, radially symmetrical animals, so radial symmetry, two germ layers. And you can see those color-coded here as yellow, in yellow and blue. The yellow is the endoderm, and the blue is ectoderm. And this is the agreed on common color coding that you'll find in freshman textbooks and in the models that are utilized for labs where this is talked about. So the endoderm is going to form the lining of the gut, the archenteron, or the gut cavity that's going to form here. Now, radially symmetrical animals only have a single opening into their gut that serves as both as a mouth and also as a waste excretion opening. So the development here is a little bit different. You can see that the early archenteron travels across the blastocele of the blastula, but it doesn't actually merge with the ectoderm on the other side. So you don't create a second opening. So we only have a single opening, and that is functionally the equivalent of a blastopore. So radial symmetry is directly correlated to the presence of two germ layers, the ectoderm and the endoderm, that form up during gastrulation. When you come to bilateral symmetry, oh, excuse me. You have three germ layers. And so the third one is the red, and that's called mesoderm. So mesoderm gives rise to all the organ systems that are associated with the body cavity and our auxiliary organs to the gut. So things like kidneys, the muscles that line the body cavity. So in our case, the muscles that are associated with the ribs and also the muscles that are associated with the digestive tract, those are all formed from mesoderm. The ectoderm gives rise to the skin, to the central nervous system, to the sensory organs associated with the skin and the central nervous system. It also gives rise to the enamel of the teeth. So there are some odd features that are associated with ectoderm. There are some odd features that are associated with mesoderm. The endoderm is pretty straightforward. It is the lining of the gut, and it also gives rise to the organs associated directly with the gut, not the auxiliary organs, but those organs that are directly associated with the gut. So bilateral symmetry, three germ layers, and we say that they are triploblastic. Radial symmetry, two germ layers, they are diploblastic. So what is the nature of protostome versus deuterostome? Well, if you know your Latin prefixes, which unfortunately we don't teach in school much anymore, proto means one, deutero means two. Stome, well, you learnt about stomata in plants. A stomata is an opening into the leaf. So stomata comes from the same Latin root. It means opening. So a stome or a stomate, to use the full extension, protostomate and deuterostomate. So stomate refers to an opening. Proto means the first opening. Deuterostome means the second opening. And what it refers to is which of the two openings becomes the mouth of the archenteron in the primitive gut, and therefore the mouth of the organism ultimately, because the archenteron will develop into the full digestive tract at some point. So in protostomes, the first opening, the blastopore, becomes the mouth. The second opening becomes the anus. In a deuterostome, the first opening becomes the anus and the second opening becomes the mouth. It is the fate of the blastopore that determines whether you are a protostome or a deuterostome. And remember again, deuterostomes are considered to still be a valid clade. Protostomes are not a valid clade. They're no longer considered to be taxonomic. They've been split up into two new clades, which we'll talk a bit more about when we do chapter 32. So that gives you the basics of gastrulation. So if we want to look at a chart that lays out which organ systems are associated with which of the three germ layers, this is a good one. It comes straight out of your textbook. This is one that you should be familiar with. It gives you basically the major organ systems that are associated with each of the germ layers. There are more, but these are the major ones, and you should be familiar with all of these. So let's take a look at the basics of a sea urchin in a little bit more detail. You've seen most of what you really need to know, but this just gives you a little bit more detail. So gastrulation for the sea urchin begins by cells at the vegetal pole migrating inwards, and those become what are referred to as mesenchyme cells. They will actually become part of the mesoderm. They grow extensions called philopoda, you can see those here, that will anchor the developing archenteron to the ectoderm on the other side of the blastocele as the archenteron develops, and pull it over to merge with the ectoderm as the archenteron extends. Some of those cells will remain associated with the developing gut, and they will form the mesoderm that will give rise to the interior body organs that are not associated with the gut. Other cells will remain at the vegetal pole and form an invagination that will eventually elongate and give rise to the archenteron, the primitive gut. The cells that remain on the outside become the ectoderm. So it's a pretty straightforward series of steps. When we look at gastrulation in a frog, it's a little bit more complicated because number one, we have to take into account the offset of the blastocele. And number two, we also have to take into account the presence of the yolk. And number three, we have to take into account the fact that the yolk is going to provide nutritive support, so the archenteron will not be connected to the exterior. It will remain locked away because the embryo is going to utilize the resources of the yolk. So gastrulation begins by some cells from the animal hemisphere migrating in down and forming a slit that is the beginning of the blastopore. The region that is dorsal to that slit, so the cells form a slit and start moving inwards, elongating the slit inwards. The outer lip on the upper surface of this slit is referred to as the dorsal lip of the blastopore. That turns out to be extremely important because it is going to mediate the formation of the dorsal ventral orientation of the animal, because it establishes the dorsal ventral axis of the animal. So these cells migrate inwards. Some of them are going to form mesoderm. Some of them are going to form the endoderm that line the gastrovascular cavity. Oh, sorry, the archenteron. I'm confusing terms here. This is the archenteron. Gastrovascular cavity is in the radial symmetrical animals. So let's just clear that one up. I didn't give you the name, but that's what it is. It's a gastrovascular cavity is the gut that contains a single opening that serves as mouth, both mouth and anus. I probably should have explained that earlier. So we have the developing archenteron. So we have this extension of cells that have migrated inwards from the animal hemisphere that are developing into both mesoderm and the lining of the gut, the endoderm. Cells from the animal hemisphere continue to elongate the ectoderm on the outside. The blastopore starts out as completely enclosing or completely encompassing all of the vegetal hemisphere. It eventually develops to encompass all of the vegetal hemisphere. But these migrating cells coming out of the animal hemisphere continue to move around and shrink the size of the blastopore. Eventually, they meet at an area that is somewhat adjacent to where the vegetal pole used to be, and a small amount of blastomeres containing yolk occupy this space and form what is referred to as the yolk plug that seals up the blastopore. Immediately underneath this ectoderm layer is this elongated layer of mesoderm. You then have the archenteron lined by endoderm, and notice that the blastocele has completely vanished. The blastocele makes no contribution to the archenteron or to the body cavity that will ultimately develop. So the blastocele is strictly the cavity found in the blastula. It makes no contribution to the archenteron. It makes no contribution to any body cavity that may develop during later development of the embryo, which in most animals does take place. We'll talk more about that on another day, actually, when we get to chapter 32, which we'll do next time. Gastrulation in the chick is somewhat different because the blastula, remember, is not a blastula. Remember, that thing that we call the yolk of a chicken egg is actually the egg cell jammed full of yolk material. And somewhere on the surface is the little disc of cytoplasm in which the cleavage process takes place, giving rise to the functional equivalent of a blastula, referred to as a blastodisc. It is made up of two layers of cells, the epiblast and the hypoblast. So when gastrulation begins in a chick egg, in an amniote in general, so chickens, reptiles, and mammals ourselves, because we are amniotes as well. We just don't lay our eggs, we retain them in the body of the female. So when gastrulation begins, cells from the epiblast begin to migrate through a structure called the primitive streak. Now, the primitive streak does not open up across the full width of the blastodisc. Instead, it starts at one end and it migrates across the blastodisc. And that establishes the anterior to posterior axis. So the formation of the primitive streak establishes the anterior to posterior axis because the end at the side of the disc at which the primitive streak first opens up represents the anterior end, and the direction in which it travels is towards the posterior end. It also establishes the dorsal ventral axis. So I'll put in here also establishes dorsal ventral. So dorsal is on the epiblast side. I'm trying to think how I'll do this. Let's see. So dorsal to ventral goes that way. Now, if you notice what's happening here, cells that are migrating inwards, some of those cells migrate into the space of the blastocele. They're going to become part of the interior mesoderm. Some of the cells will remain in what was the epiblast, also to give rise to mesoderm cells. Some of the cells will migrate completely across and displace the cells of the hypoblast. Those are going to become the endoderm, the lining of the gut. So one of the things that we need to recognize here is that gastrulation in amniotes involves cells from the epiblast only. So the hypoblast is not going to make any contribution to the embryo because the embryo is going to develop from these three germ layers. So the displacement of the hypoblast cells by the epiblast cells migrating completely across to form the endoderm is an indication that the hypoblast is no longer contributory to the embryonic cell mass. And this is something that we notice in amniotes very specifically. So cells in the blastocele in the hypoblast let me rephrase this. Cells in the hypoblast as components of that functional equivalent of the blastula, the blastodisc, they are being eliminated from participation in development of the embryo. This is something that is somewhat unique to amniotes, the fact that not all cells in the blastula are going to contribute to the embryo because they get moved out of the way as part of gastrulation. As the germ layers form, they are not formed by all of the cells contributing to the blastula as we've seen previously. So this is a difference as far as amniotes are concerned. So in humans, gastrulation takes place once the cell mass that is developing from the zygote makes its way down through the fallopian tubes and enters into the uterus and ultimately implants in the uterine wall. Only then does gastrulation take place. So it's delayed by about 7 to 10 days post-fertilization because it takes about that time for the developing early embryo to make its way out of or down the fallopian tube where fertilization actually takes place, and the early steps, the cleavage process takes place, down through the fallopian tubes to enter into the uterus and then ultimately to embed or implant in the wall of the uterus. And even at this point, we are going to see that there is a discrimination between cells that will participate in the formation of the embryo and cells that won't. So let's take a look at the diagram. So remember again that fertilization takes place up in the fallopian tubes. This is one of the reasons why such a large number of sperm cells are found in the male ejaculate. In a human, it's roughly 150 million individual sperm cells. The vast majority of those are going to die either in the vagina or in the uterus. Very few of them are going to make it across the uterus and up into the fallopian tubes. It could be measured in the few thousands. And it's not the most rapidly swimming sperm cells that are actually going to do the fertilization, because there is a process called capacitation that takes place in the fallopian tubes that preps the sperm cells for the fertilization step and gives them the ability to make their way through the follicular cells to get access to the egg cell. And those sperm cells that race ahead do not get capacitated and therefore can't make their way through the follicular cells. It's the sperm that take a little bit of time that undergo capacitation and actually do fertilization. That takes place up in the fallopian tubes. As cleavage takes place, cells start to separate out into the blastula. And the blastula itself may contain about 100 cells in total, and it's made up of two distinct sets of cells. There are the cells that form the outer layer of the blastula referred to as the trophoblast. Those cells are now excluded from forming the embryo. The only cells that will contribute to the embryo are the 20 or 30 cells that are sitting inside the trophoblast called the inner cell mass. Those are going to participate in the formation of the blastodisc, and then subsequently participate in gastrulation. But again, not all cells of the inner cell mass are going to participate in gastrulation, because remember, we're dealing with an amniote. And in amniotes, the blastodisc is composed of the epiblast and the hypoblast. And just as we saw in the chick, the cells of the hypoblast are going to be excluded. So only epiblast cells migrate during gastrulation. Some of them start the formation of the ectoderm that will form the outer layer of the embryo. Some of them migrate out and form the mesoderm, and some of them migrate further and form the lining of the gut cavity, which obviously will not be a functional gut cavity, because the placenta is going to be involved in moving resources from the mother to the embryo, and the mother will just carry out all the digestive processes. The baby will simply take benefit of that because of the transport of gas and nutrients from the mother's blood into the embryo. The placenta itself is formed by tissue derived from the hypoblast, tissue derived from the epiblast, and tissue derived from the trophoblast. So all three of those structures contribute to formation of the placenta. In the meantime, also developing are the four extraembryonic membranes, what are referred to as the chorion, the amnion, the yolk sac, and the allantois. Now, in placental amniotes, that is mammals, those membranes serve substantially different functions to the original functions that they served in egg-laying amniotes. So let's take a look at the function of those membranes in egg-laying amniotes, because there we see the functions that they were intended to serve. The functions that they serve in placental amniotes, or in mammals in general, not placental amniotes, I should say mammals in general, because we also need to include their monotremes. Well, monotremes are egg-laying mammals. So marsupials, which are not placental mammals, but are mammals nonetheless, and are amniotes, and then placental mammals like ourselves. The role of these extraembryonic membranes is a little bit different in non-egg, I'll term it this way, non-egg-laying amniotes. That's probably a better term to use. So let's take a look at their role in egg-laying amniotes. So as I mentioned, the four extraembryonic membranes are the chorion, which is involved in gas exchange. So if you ever get an opportunity to look at video of a chick egg that's had a hatch cut in the egg cell, in the eggshell, which is how we manipulate chick embryos. We do it under sterile conditions. You can actually see gas bubbles rising from the chorion, which indicates that its role is gas exchange. The amnion, which is a membrane, forms the amniotic sac, which is the fluid-filled sac that completely encloses the embryo and protects it against accidental damage as it moves around. It also means a connection back to the aquatic origin of all life, because it is a salty fluid. It kind of resembles seawater. So even though amniotes are able to reproduce away from a body of water, they still carry a vestige of that in the form of the amniotic fluid inside the amniotic sac. So essentially, all animals complete their early embryonic development in a fluid environment, whether it be in water itself, either fresh or marine, or in a small remnant of an ocean in the form of the amniotic fluid. The yolk sac in egg-laying amniotes completely encloses the yolk and is the source of some of the very first blood vessels to develop that are involved in transporting the breakdown products of the yolk nutrients to the embryo. And so the yolk sac mobilizes the contents of the yolk and allows it to be transported to the developing embryo for its use. Now, because in egg-laying amniotes, the development is taking place in a sealed space, whether it be a reptile in a leathery shelled egg or a bird in a calcium carbonate reinforced eggshell, you can't dispose of the waste products, and specifically the nitrogen waste products, which are toxic to the organisms. That's why we have kidneys to filter out that nitrogen waste and eliminate it through the urine. But you can't dispose of it that way if you're enclosed in an eggshell. So you need another space in which to enclose it, and that's what the allantois does. It's where the nitrogenous waste, the product of breakdown of amino acids, is disposed of, because that is toxic to the organism. And as you can see, it also contributes to gas exchange. Some of these membranes also contribute partially to the formation of the placenta, which shouldn't come as a surprise, because remember, the placenta is carrying out all of these tasks in placental mammals. And so the placenta does gas exchange, the placenta moves the nutrients from the mother's blood to the developing embryo and takes the waste back to the mother's blood so it can be eliminated. So the chorion, the yolk sac, and the allantois also contribute somewhat to the development of the placental from the embryo side, not from the mother's side, but from the embryo side. So here you can see those four membranes arranged inside, in this case, a chick egg, but this would be typical of a egg-laying amniote. So once gastrulation has taken place and you've got the three germ layers in place, the next thing is the beginning of organogenesis. And this is going to give rise to very primitive, but nonetheless somewhat functional organ systems that are going to support the development of the embryo, mature in their own right, and enlarge in their own right as the embryo enlarges, so that they reach a point at which they can become supportive of the independent young, whether it be an independent young in some sense, independent of its mother's support in some fashion, or independent of support via the yolk inside a shelled egg or something like that. So organogenesis usually begins with the formation of the neural tube and the overlaying neural plate that is going to cover the underlying neural tube. So if we take a look at a frog as the model organism for vertebrates, we see that the ectoderm tissue that overlays the position that will be occupied by the neural tube and subsequently the central nervous system. So this is a dorsal orientation. You see that cells that formed a single layer start to change their shape, migrate towards each other, and then elongate to form a single layer of what we refer to as columnar cells that once represented a space occupied by two layers of cuboidal cells. In the process of taking on this columnar orientation, they start to form an invagination, a folding and a rolling that takes place here. And then on the edges adjacent to the unmodified ectoderm tissue, you get this region that is referred to as a hinge region, where you actually get a reverse folding. So this folding is going this way, this reverses the folding to go this way. As this folding continues, the neural tube forms up as a completely enclosed tube, and over the top of that tube are a layer of cells that are referred to as neural crest cells. These are also derived from ectoderm tissue, hence the blue coloring. The neural tube will become the dorsal nerve cord that is characteristic of vertebrates, and will be protected by bony material that becomes the vertebral column, the vertebrae of the vertebral column. The neural crest cells give rise to some of that. They also migrate forwards and give rise to parts of the skull that is a key feature of vertebrates. The vast majority of vertebrates have at least a covering over the brain, a cranium, and vertebrates that have a more complete covering of the brain and a more obvious head actually have a complete skull with jaws and all of those features that are associated with it. Some of those features are derived from the neural crest cells. They migrate forwards to give rise to that. Underneath the developing neural tube, you get a condensation of mesoderm tissue that also forms an anterior-posterior structure that will become what in vertebrates and chordates, remember chordate is the phylum, vertebrates is a subphylum within chordates, will become a structure known as the notochord. In chordates, this remains a cartilaginous structure, but in vertebrates, and particularly in vertebrates that have a bony skeleton, which is the vast majority of them, the notochord or the cartilaginous portion of the notochord gets displaced by calcium carbonate crystals that are secreted by osteocytes, the living cells of bones. There is always a remnant of the cartilage left behind, and if you're looking for the cartilaginous remnant of your notochord, think about the cartilaginous discs that separate the pairs of vertebrae. Your vertebrae actually don't rub on each other. That would be incredibly painful. Ask anyone who's had a breakdown of their discs just how painful that is. So we have small cartilaginous discs inserted in between or remaining in between each pair of vertebrae so that the bones don't rub on each other. They are derived from the notochord. The mesoderm itself will give rise to the musculature, the interior musculature, depending on the type of body cavity that is present. The layers of muscle, there could be a single layer of muscle or there could be two layers of muscle. If there's a single layer of muscle, it'll be one on one side of the body cavity. If there are two layers of muscle, it will be on both sides of the body cavity. And that in turn determines what type of body cavity you're looking at. The origin of that body cavity, in turn, if it's a true body cavity lined by two layers of muscle, also determines what type of organism you're looking at. We'll talk a little bit more about that in chapter 32. What you should also bear in mind is here that the gut cavity, so here you can see the archenteron, or if we were talking about a functional gut, which this is not in the frog, which has a mouth and an anus. So that would be a true gut. That gut cavity does not count as the body cavity. So you're going to see when we start talking about the animal kingdom that some of the basal animals, some of the basal bilaterals, lack a body cavity completely, even though they have a gut cavity. The gut cavity does not count as a body cavity. They are separate entities. When you look at gastrulation in an insect, for instance, in a Drosophila, which is in fact what we're looking at here, the formation of the neural tube still is the first. That can't be right. The way that is labeled is not right for a Drosophila. And I'm just remembering something that was indicated in the textbook as I was reading the textbook. So bear with me for a minute. I may have noticed something that you really need to be aware of, the way this is labeled. Let me take a look at this. Because that's not right with the way that's labeled, if that is. What are they labeling this at? Okay, so they are labeling it as a frog embryo, because it is a frog embryo. How can I tell? So I'm not sure where the Drosophila idea came from. It may have been because this was labeled that way in a previous version of the textbook, or maybe I'm misidentifying the diagram that I'm looking at. I think that may be the case. Anyway, this is a frog embryo, and the reason why I know is because it's got a notochord, and a notochord is characteristic of a vertebrate. So this has to be a vertebrate. So yeah, this is a frog embryo. The point of looking at this is to show you what happens when the muscle tissue starts to condense out. So this you can see here as these, I'm trying to think of what the right word to use here is. Segments of muscle, yeah. And that is the correct term. So let's say these segments of muscle, which are referred to as somites. One of the things that you notice in vertebrates, and you notice this in a lot of insects, and you notice this in a lot of terrestrial vertebrates, is that the body is segmented. So if you're looking for your own evidence of segmentation, one way is to look at the vertebrae. So the vertebrae show segmentation. The ribs that are associated with the vertebrae are evidence of segmentation. And then the musculature that is associated with the ribs is evidence of segmentation. Now, one of the other evidences of segmentation, which some of you may have, I do not, is the so-called six-pack abs. If you look at those layers of muscle in someone who has a well-defined abdominal musculature, that is your evidence of segmentation there, the layers of muscle that run down the abdomen. And they are all derived from this condensed mesoderm that gives rise to these segments of muscle, which in the early embryo we refer to as somites. And you're going to see these turning up. You should have seen them in some of the pictures that you looked at in the embryology lab. You're going to see these turning up in a couple of other pictures that we will look at later in the presentation. So yeah, I'm not sure why I was thinking this was a Drosophila. There is another picture, and maybe that's the one that I'm confusing here. When I look at this EM picture, it is similar to an early Drosophila embryo, but I should have picked up on the somites. That's my mistake. Organogenesis in the chick as an example of a terrestrial vertebrate is very similar in that you get the neural tube forming first, the notochord condenses out underneath it, mesoderm condenses into the skeletal muscle, and you get the somites forming. The yolk sac, of course, is a feature that you're not going to see in a frog, because here we're talking an amniote. So the yolk sac is going to be in contact with the yolk to process the yolk material, but then it forms this stalk that connects into the embryo to mobilize the yolk products to support the developing embryo. And you can see some of the structures starting to form up here in the chick embryo. So this would probably be at around being late organogenesis, this is probably going to be around 96 hours is what I'm going to guess. I don't know what the labeling is on this. I could look it up, but this is going to be around 96 hours post-fertilization. And you can see that it's already pretty well developed. You can see the neural tube. You can see the somites. It's got an eye. The brain is starting to develop. It's got a functional heart. Here are the blood vessels coming in from the yolk to mobilize the yolk products into the embryo to support its growth. Now, you're not going to see all of these features in a human embryo at the same stage. Our development takes place a little bit more slowly. So whereas this might be around 96 hours in chick development, this would probably be more in keeping with around 6 to 8 weeks in a human embryo development. It starts to take on these sorts of features. Remember, by the time we get to the end of the first trimester, which is 12 weeks post-fertilization, basically all the body organ systems are in place. They're likely to be extremely primitive, but they are all present nonetheless. So one of the important things to understand about both organogenesis and gastrulation is that there is a lot of movement of cells. The cells are moving from one place to another, which we refer to as crawling. They're also going to be changing shape, and we've already seen that in the formation of the neural tube. I mentioned the idea of cuboidal cells. They're square shape, turning into columnar cells. They're more rectangular in shape. We're going to find that cells are going to start sticking to each other, both locally, but also as a consequence of crawling from one location to another. They actually attach to each other once they reach their final destination. So there's a lot of things going on here that we need to have some way of explaining. And so when we start thinking in terms of change of shape and we start thinking in terms of moving around, we have to think in terms of what sorts of changes are going on inside the cell with respect to the cytoskeleton, because the cytoskeleton is the internal structure of the cell that causes the cell to change shape. And so we can see reorganization of the cytoskeleton exemplified in the formation of the neural tube. So here are the cuboidal cells that are in the ectoderm. So let's label this. These are cuboidal cells in ectoderm. And you can see that in those cells, the cytoskeletal components have a very loose arrangement, no really developed organization. As the formation of the neural plate begins, we see the transition of these cuboidal cells into columnar cells. That elongation into columnar cells is driven by microtubules growing. At one end of the cells, and in this case, it's the outer end of the cells that are facing outside of the embryo, a layer of actin filaments, microfilaments, forms up at right angles to the microtubules. Once the elongation of the microtubules has completed, the microfilaments contract, as microfilaments do, and that narrows this end of the cell and broadens the other end of the cell. And that's what leads to the rolling around of the developing neural tube. At the hinge region, you see the same thing happening, but this time it's the interior end of the cell that contains the microfilaments that contract to narrow that end. And the microtubules have elongated the cell outwards, and that's where it fans out and broadens rolling, creating a roll back in the other direction at the hinge point. The continued rearrangement of the cytoskeleton in all the cells that will ultimately compose the neural tube give rise to the completed neural tube. Cells from the ectoderm then break off and migrate inwards to form the neural crest cells that you can see here. Notice they're still cuboidal in shape. So the formation of the neural tube is nothing more than rearrangement of the cytoskeleton to create a tubular structure from cuboidal cells. It's all about rearrangement of the cytoskeleton. A note here, formation tube. Is a consequence of cytoskeleton rearrangement. So it's simply just simply the cytoskeleton rearranging the shape of the cells that leads to the formation of the neural tube. At the same time, however, we also need to recognize that cells crawl, and crawling can be in two forms. One could be literal migration of the cells from one location to another, which we've talked about a little bit in terms of gastrulation. But it could also be movement of cells to rearrange the layers of cells into a tissue into something different. So there is a process called convergent extension that is included under the umbrella of cell crawling that really is a rearrangement of cell layers into from one form of layering to a different form of layering. So here are cuboidal cells arranged in two layers. And this may very well be the origin of the cuboidal cells that are forming the neural plate that will ultimately fold to give rise to the neural tube. A similar sort of process almost certainly takes place. So the two layers of cuboidal cells start to elongate and crawl towards each other as a consequence of this elongation under the control of the microtubules. They crawl in between each other, a process called intercalation. So you have this cell moving in between these two cells. This cell is moving in between these two cells, and likewise. So this is what is meant by intercalation. They insert themselves in between each other as they elongate. Then once they have fully grown in between each other, then the cells expand sideways. There's the extension to increase the span of this tissue layer. So you're not actually creating new cells to go from here to here. What you're doing is rearranging the internal structure of these cells to create an elongated layer of cells. And we know this sort of thing is involved in, for instance, the elongation of the neural tube, where cells tend to migrate to the center of the dorsal axis. And so they intercalate in between each other, and then they elongate to extend the neural tube and elongate it along the dorsal ventral, along the anterior posterior axis of the organism to create a neural tube and a notochord that span the anterior posterior axis of the developing embryo. So convergent extension is a form of crawling, and it is a way of relocating the cells with respect to each other. But then we also do know that cells do literally walk from one location to another, or crawl from one location to another. And what allows them to do this is the extracellular matrix of the cells that provide the crawl paths or crawl tracks that they're going to migrate along. And so what we've discovered, what's been discovered, is that the cells that surround the group of cells that are going to migrate send out signals that trigger the cells to begin their migration. As they walk along the extracellular matrix, the cells that surround the crawl path also send out chemical signals to encourage them not to deviate from their crawl path. And then the cells that are located at their destination also send out chemical signals that act as attractants. And so the cells are guided from their original location to their destination by chemical signaling from cells that are adjacent to them. And this provides them with a pathway so that they wind up in the correct location. When they get there, surface proteins act as cell-cell recognition proteins and also act as cell-cell adhesion proteins so that they can bind up with each other if that is necessary, which it typically is. Now, if you interfere with the extracellular matrix, which you can do, you can block the extracellular matrix, you can disrupt the ability of the cells to migrate, and what you wind up with is a disorganized mass of cells that no longer form a nice, neat layer of cells because they have no clue as to what they're supposed to do. They're not receiving the signaling. The matrix is not providing them with the signals that they need in order to be able to crawl from one location to the next. So they simply form a disorganized mass of cells. Then, in terms of adhesion, when the cells get to the right location and adhere and bind to the cells at their location, we have a large family of cells called cell-to-cell adhesion proteins that are responsible for this. And one of the subgroups of cell-cell adhesion proteins are a group of proteins called cadherins. And these are important at various stages during the development of the embryo. And the particular cadherin that I'm going to show you the experimental results for is one that's involved in forming the blastula and then obviously from there, subsequently the gastrula. So let's take a look at what happens. So here is a control embryo with fully functional versions of the particular cadherin that's involved here, known as EP cadherin. So this embryo has formed up, developed normally. The blastula is in good shape. Gastrulation will occur normally. That's our expectation. Here is the embryo in the absence of this cadherin. And the way this was prevented was by injecting the egg cell that was used to form this embryo with an antisense RNA molecule that bound to the messenger RNA for EP cadherin when it appeared through base pairing. So it was oh, I forget the right term now. So much terminology. Anyway, I'll probably remember the term, like the proper definition. But base pairing is what is the mechanism for blocking the synthesis of EP cadherin, because the messenger RNA binds up this little interfering RNA strand, and that prevents translation. The protein is not produced. And so we've just prevented the appearance of a single protein, the EP cadherin, which is responsible for the correct binding of some of the cells that are involved in the formation of the blastula. And you can see what's happened here. The embryo is completely disorganized, and this will not complete gastrulation. In fact, it probably won't begin gastrulation. There's no way that this embryo could complete normal development. And so this reinforces the fact the binding of cells to each other in the correct pattern is also part of embryological development. And we're going to see more of that in a few minutes as we continue talking about the development of the embryo. But first, let's take another break. And again, I will shut down the recording so that you can take a break, but I will see you back on the other side with the answer to this concept tech question. What would you expect to happen if you block the function of microtubules in frog embryos before the neural tube forms? See you on the other side with the question, with the answer.